Bread is one of the most widely consumed food in the world. For sure, we have experienced having our bread spoiled because of certain organisms, such as fungus. Therefore, we humans have made ways to preserve our bread through different preservatives. However, preservatives, especially artificial ones, are not safe for our body. And the viable option on the world today are artificial preservatives, which are being used in many ways. In order to solve that, we need to consider making and using natural preservatives. Now, allow me to introduce ourselves. My name is Andrew Reinhardt, and today I'm joined by Gabriel Fabiano and Kresna Jenny to present our research on the antifungal effect of rhubarb petiole and banana peel against bread spoilage fungi growth. To start off, we would like to state our reasons why we have chosen this specific problem to solve. Food waste, as we know, is currently one of the main issues that the world has to solve. In fact, based on the United Nations Food and Agriculture Organization, every year, 1/3 of the food in the world are being wasted. Based on research, spoilage is actually one of the biggest reasons why. Now, out of every organism that causes food spoilage, fungi are one of the biggest contributors. With bread being one of the most widely consumed food in the world, we are trying to solve the problem by considering the impact that we can contribute. The problem lies in the preservative itself. The preservatives that we use right now are artificial, which contains chemical substances that are not safe for our body. Therefore, natural preservatives are very much important in the long run. Based on the research of previous researchers, rhubarb petiole is a viable option in preserving food due to the existence of rain and amodin within the petiole, which are in fact an antifungal substance. Another viable option that we consider is banana peels that contains flavonoid, which are another substance that are antifungal. Therefore, based on those two research, we decided to use both of them as our main treatment to the bread. This research has two purposes. The first goal is to study the antifungal effect of rhubarb petiole and banana peel against spoilage fungi growth of bread. And the second one is to study the optimal percentage of rhubarb petiole and banana peel to minimize the spoilage fungi growth on bread. Now, the presentation will be continued by Gabriel Fabian.
Thank you, Enrel, for the introduction. I will continue by explaining the basic theories of this study. First is rhubarb petiole. Rhubarb is defined as a vegetable that comes from cultivated plants in the genus Rheum and the family Polygonaceae. Petiole is defined as the stem of a plant, making rhubarb petiole the stem of the rhubarb plant. Generally, rhubarb is used in foods such as rhubarb pie or traditional medicines, mainly originating from China. As mentioned before in the introduction, rhubarb petiole's antifungal properties are derived from the phytochemical it contains, which are rein and amodin. Rein, which is also known as cassic acid, is defined as a substance in the anthraquinone group that is obtained from rhubarb. Anthraquinone is an organic aromatic substance with the formula C14H8O7 with a ketone group in the central ring. Rein can be found in rhubarb plants, including the species used in this research, which is Rheum rhabarbarum. A research titled Phytochemical and Antifungal Activity of Anthraquinones and Root and Leaf Extracts found that rein inhibits fungal growth significantly, as it is antiseptic and toxic to fungi. Another antifungal anthraquinone from the rhubarb petiole is amodin, or 6-methyl-138-trihydroxyanthraquinone, which is a chemical compound that can be isolated from rhubarb. Research have shown that amodin derived from plants serves as a fungicide and fungostatic agent that inhibits the growth of various types of fungi. This antifungal activity is found to be caused by changes in the cell membrane permeability from lipid changes in the membrane. Aside from rhubarb petiole, we will also be studying banana peel's antifungal activity. A banana is defined as an elongated edible fruit produced by several large flowering herbaceous plants in the genus Musa. Banana peels have many functions outside of its biological purposes in protecting the banana fruit, which are used as livestock food, in water purification, as well as ethanol, cellulase, and compost production. Banana peels contain flavonoids, which is found in a research by Adayanti. Flavonoids are defined as a group of natural compounds with variable phenolic structure. Moreover, studies have shown that flavonoid compounds have antifungal mechanisms by interfering with mitochondrial homeostasis and also by disrupting the integrity of fungal cell membranes. Next is bread, which is defined as a usually baked and leavened food made of a mixture whose basic constituent is flour or meal. In this research, we will be using plain bread according to the AACC method, which will be discussed in further detail in the method section. Lastly, bread spoilage fungi and their growth. Bread is made of starch, which causes rapid growth of mold on bread. Bread's general texture also makes it easier for mold to grow, which are damp and or wet conditions. These microorganisms decrease the quality of bakery products due to its contamination, in particular because of mycotoxins produced, which could cause fever, inflammation, shortness of breath, etc. if consumed. Common types of fungi found on bread are from the genus Aspergillus, Penicillium, and Rhizopus. Next, my colleague Kresna will be explaining the methods used in this research.
Thank you, Ian. Now allow me to show our methods of experiment. First, we have our variables. The independent variables include the rhubarb petiole and the banana peel. And the dependent variable is the fungi growth, which we will analyze by comparing the percentage of the fungi surface area to the bread and by identifying the most common fungi types that will grow. Next comes the experimental design. Our bread preparation methods were based on the approved methods of the American Association of Cereal Chemists, or AACC, and our fungi identification methods were based on the introduction to food-borne fungi. We analyzed 7 different bread group treatments. The first treatment, bread group 0, or BG0, had no rhubarb petiole and banana peel. This is the control bread group. It acted as a base for the rest of the other treatments. Now, bread group 1 contained approximately 3% of the rhubarb petiole. Bread group 2 contained approximately 4% of rhubarb petiole, with no banana peel. For the bread group 3, contained approximately 3% of banana peel. Bread group 4 contained approximately 4% of banana peel. Now, for bread groups 5 and 6, these were a combination of the rhubarb petiole and banana peel. Bread group 5 contained 1.5% of rhubarb petiole and 1.5% of banana peel, so each with 1.5%. Lastly, bread group 6 contained 2% of rhubarb petiole and 2% of banana peel. We repeated each treatment 4 times as calculated from the Federer formula. Our bread ingredients, according to the AACC, as I've mentioned, consisted of 250 grams of flour, 15 grams of sugar, 5 grams of salt, 10 grams of oil, 3 grams of yeast, 1.75 grams of bread improver, and sufficient water. Now, we have created an illustration. So from this animation, you can see the first step when the dough was just formed. Next, we have the rhubarb and the banana peel. They were chopped into tiny pieces, which allowed us to put them in each separated dough according to the bread group treatments. Then the bread was baked, cut in half, and put into plastic bags. The last steps was to play the waiting game, patiently waiting for the fungi to grow. Now the experiment took place at Pulau Pantara 4 and 4 number 11 from the 20th of December to the 29th of December 2020. Here you can see the bread before they were baked on the left and after they had been baked and put into plastic bags on the right. Now our data analysis methods. We analyzed our data using 2 methods. First, we analyzed the fungi identification and growth time. This one were carried out microscopially according to the Samson method and measured for the elapsed time period until it is visible. The second method was by calculating the surface area. This was carried out digitally by calculating the visible area of the bread's spoilage fungi compared to the surface area of the bread.
Now I'm going to discuss about the results. So based on our analysis on the fungi identification and growth time parameter, as you can see from the picture, there are three main fungi groups. The first one is the A fungi group, which consists of the Rhizopus genome, especially Rhizopus thorni per species because of the back colony. The B fungi group, the Penicillium genome, especially Penicillium italicum and Penicillium digitatum species because of the white colony. And on the C fungi group, the Aspergillus genome, especially Aspergillus fumigatus, Aspergillus niger, Aspergillus flavus species because of the green or yellow colony. Now, based on the analysis before, the fungi group has each own growth probability for each treatment. So, based on the graph, you can see that the group A, group B, and group C have showed different kind of results. Now, with the addition of banana peels on the bread group 1 and the bread group 2, it reduced the probability of growth of fungi group B, as you can see on the graph. And for the second one, the addition of rhubarb petiole at a higher concentration at bread group 4 drastically reduced the probability of growth of fungi groups A and B. Now, this result showed that the higher percentage of rhubarb petiole and or banana peels leads to a greater antifungal effect on the possible growth of each group of fungus. Each additional percentage of rhubarb petiole and or banana peel shows two kinds of results: did not affect at all or decrease the probability of growth on each type of fungal group. However, there's an exception on bread group 3 to bread group 4 with the additional 1% of petiole rhubarb from 3% to 4% in the group C fungi did not affect at all. Now, this indicates that the antifungal effect of petiole rhubarb decreases once it passes its optimal percentage. Now, this part will be continued by my colleague, Gabriel Fabian.
Thank you, Enril. Our next parameter of measuring bread spoilage fungi growth is surface area. For calculating bread spoilage fungi surface area, we used computers to measure the area of the visible fungi, which is colored red, in comparison to the total bread surface area in percentage, which is the sum of the red and the blue area. This is the visualization of our data. The results shows that the antifungal ingredients from rhubarb petiole and banana peels are effective in inhibiting fungal growth on bread, with the most effective result being the addition of 4% banana peel in bread group 2. On average, this treatment was able to reduce fungal growth by 29.75% compared to the untreated bread group. The second most effective antifungal agent was the addition of 4% rhubarb petiole at bread group 4, which reduced fungal growth by 28.53% compared to the control. As seen from this graph, the higher the percentage of rhubarb petioles and or banana peels, the greater the antifungal effect on the surface area of fungal growth. This was found in each treatment, rhubarb petioles, banana peels, and both. Next, Kresna will conclude this research presentation.
From this experiment, it can be concluded that the rhubarb petiole and banana peel have an antifungal effect on preventing fungal growth on bread. We can pull two conclusions from the two methods. The best inhibiting effect of fungal growth in terms of the probability of mushroom growth was found in the addition of 4% petiole rhubarb or bread group 4 treatment. The best inhibiting effect of fungal growth in terms of surface area was found in the addition of 4% banana peel or bread group 2 treatment. This treatment is effective for both measurement of surface area and probability of fungal species. Now, the research team hopes that this research can be developed by examining the method of preparation of the rhubarb and banana peels to optimize their antifungal effect without affecting the taste of the bread. Thank you. That's all from us. We hope you like this presentation. See you in the next one. Thank you.